We are going to be talking today about my favorite topic, biotechnology. It is how my current supervisor lured me away from my previous district, and it's now what I teach. And it's a fantastic topic that I am lucky enough to be able to get to teach, because a lot of times when we think about what it is in a CP bio or an honors bio, even an AP bio, it's not an afterthought, but it's one-off lectures. It's a topic that you maybe get to cover at the end of the year. It's something that unfortunately you don't get to go so in depth on. We end up then teaching to a test, whether it's our test or College Board or a different governing body. We teach to that test, and we hope for the best. One of my colleagues and I at Livingston, we say that we are very good at raising owls, such that they take in that information and they get really good at taking it in and barfing it back out to us. And what I want to do with what you can do with this topic, with this kit, is really change that into something that we're going to let the kids master something. Because when you think about our landscape today, biotechnology is at the heart of the medical industry and absolutely beyond. And think about this next statement here for a second. Our high school courses might be the very last time one of our students takes a lab science course. Scary, very scary, especially in today's landscape. But think about it. Sometimes, Michael, I agree with you. That is like an awful statement. And when one of my friends said that to me, it really shook me to my core. But think about it. We're jaded because we're science teachers and we love what we do, and we took all these courses in college and master's courses, and it's what we live for. But some of our students, they'll never see a science class again. So when you think of how biotechnology in just the last couple months has developed different assays for detecting viral genomes, it's made viral proteins, and it's being able to reformat them to make vaccines, and all of this amazing technology that is coming out is all because of biotech. And so if it's that important to our future, shouldn't we make it a little bit more important to bring that into our world and into our classrooms? So enter in the One Kit Wonder. So what this kit enables you to do is it enables you to teach not one, not two, not even three. It enables you to teach 5 different topics about biotechnology that are the center of the biotech world. It enables the kids to interact with the material and to not just be an owl and regurgitate that information back to you, but actually master it. So without further ado, let's get started. The One Kit Wonder, using models to teach biotechnology concepts. Brought to you by 3D Molecular Designs, and a talk given to you by me, Diane Sigalas, the biotechnology teacher at Livingston Public Schools, part of the Livingston Biotechnology Pathway. So here's the thing about models and all of these kits. You need both of your hands. So I don't want anyone to worry about how or where to get these slides. You can either go to the link that Heather just put down in the chat, and you can get these slides directly. You can take a screenshot of this and be able to go back to them later. I don't want you to have a pen. The only thing I want in your hands later on are our nucleotides. So relax, let's play with some nucleotides, and let's see what this kit can do. So let's answer a couple of the whys. And the first one that I want to talk about is, why me? Again, my name is Diane Sigalas, and I developed an intensive 2-year biotechnology program that we use at the Livingston Public Schools, and it's become our biotechnology pathway. So in the first year, what the kids do is they interact with all of the different methodologies, and they learn how they work, and they learn how to do all the different assays. And then the second year, we dive into applications. So they'll do some pharma, they'll do some ag, they'll do virology, and then we release them off to develop their own protocols and to really do some independent-based research within our classrooms. And the modeling component within my class It's not every day, but it's pretty often. What I found that the modeling enables the kids to do is that it enables them to interact with the content in a way that I've never been able to get them to interact with it before. It enables them to really master what they're looking at. So if you look up at the top, you see my kiddos looking oh so spiffy in their lab coats. And at the bottom there, you'll see a picture of what would become the bacterial transformation lab that you can do with this biotechnology kit that we're going to talk about today. So why CBM? CBM stands for the Center of Biomolecular Modeling. It's part of Milwaukee School of Engineering. I first came in contact with Tim at NSTA in Atlanta. And Tim started talking about this science and ethics of genome engineering course. And my little ears perked up and I was like, what do I have to do to go to that? Because, I mean, that's at the heart of what I teach. So the first hurdle that I had to go through was this Modeling the Molecular World course. And I went into there thinking, okay, what am I going to learn? I've taught this stuff for years now. What am I going to learn? That was the biggest understatement I could have ever walked into something thinking, because I walked away learning so much. Their teacher professional developments are the best you can get your hands on. And what I can tell you is this. You know, we were saying teach to the test and our students are the owls. Confession time. I was always that owl. And it wasn't until that course that I actually understood how the single amino acids are going to really interact within a protein and how changing one will change everything. Did I teach it? Yeah. Did I ace those tests way back when? Yeah. But it wasn't until this course that I really understood what was going on. And since then, I have never let my kids walk out of my room without that understanding, because it's at the core of literally everything. So if you want my advice, can't do it this summer, sign up for our next summer's courses as soon as you can. You won't regret it, and your future kids will honestly thank you for it. So 3DMD is the arm that is going to develop the products that we have, that we talk about with the CBM. So CBM tells you how to use the kits and how to give you actionable ways that you can develop your pedagogy around them, and you're going to get them from 3DMD. So they have what I would call the big two, where you have the amino acid starter kit. Remember how I said I was that owl that just regurgitated? That kit revolutionized the way that I see amino acids and proteins in general. Their second kit that I would say is amazing is their flow of genetic information kit. You can teach everything, the whole central dogma, from replication all the way through to protein modeling. And what you can do is you can attach a kit that connects the two of them together. It was perfect. This was the dynamic duo of Mark and Puri 1 for biotech. And it's breaking my heart that I might not be able to do that this year. But if you come to the booth, we can talk and plan. And I've got some ideas about how I'm going to change this up for next year. But what I want to do now is introduce you to the big three. Move over, Pearsons. There's a new set of triplets in town. And here's the Biotechnology Kit. This kit enables you to make your models go so much further, go so much deeper. Because what I'm going to tell you right now is models equal mastery. These three kits enable your kids to master the content. So let's dig a little deeper into what goes into the Biotechnology Kit. And if you're no stranger to 3DMD and some of these kits, you'll recognize some of these pieces because we've seen them before. But what's magical about this combination of pieces is that it enables you to do all of those components that are on the bottom piece of this slide. So all of these things you're going to be able to go back to. This means you're pulling out one kit, not once, not twice, again, 5 different times throughout the year. enabling that mastery level to continue going. The kids get really comfortable with the pieces, really comfortable with modeling time, and they really start to excel within your classroom. The best part, these kits come packed with activities that we've already made for you. We've stepped in the cow pies, we've made those worksheets, all the storylines done, the sequences done, the questions done, connections to scientific papers in there. Everything is in there just out of the box. So all you gotta do is open it up and get ready to start that journey. So let's look at what we're going to be using today. So we are going to be talking about the Biotechnology Kit. So it's item number BTK01. What we're going to be using and what you have in front of you are the Nucleotide Student Modeling Packs. So we're going to be talking about the Biotechnology Kit, but you're going to be using and interacting today with the Nucleotide Modeling Kit. Can I show you pictures of what my kids did in the classroom? Yeah, that's easy. Could I have showed you a video on how they work through the PCR themselves? Simple. But what we're going to do is you're going to do it for yourself today, because the only way to really understand how amazing modeling is if you actually model yourself. So we're going to have some playtime. That's what my kids call it. When I take these models out, they get so excited because it's playtime and they're just ready to go. And I love the fact that they call it playtime, because that means that their guard is down. And when their guard is down, you can slip in all of that content without the fear and the anxiety that comes over our kids these days. So it is a fantastic way. So let's start playtime. Before we do that, I want to get through some workshop goals, and here are what my goals are for you today. I want you to leave this session, number one, seeing how this kit can fit into your curriculum right now. I want you to see how this kit is going to increase student learning and mastery of content and the topics that you're already covering in your curriculum. Number two, we are going to practice PCR. It is one of the 5 things that you can teach and use this biotechnology kit for. And number 3, you can tell I'm really excited about the modeling component of my curriculum. And I would really like it if you guys got excited too. So I know budgets are tight, living with that right now. And a lot of times supervisors want to see or want you to justify how this fits in with the NGSS curriculum, or if you're like Jersey and you Jerseyfy it and make it a different acronym, it's still NGSS. So what you have right here are the different steps, TRIPSes, and the DCIs that go along with it. If you need this to help your supervisor realize how important this is for you, it's right here. And what's even more is I'm going to have icons show up as we go through so that you can easily see how these steps and TRIPSes are going to be integrated into the activities that you're doing. So here's our first poll. So what we're going to do is we're going to use a new, well, I guess it's not new, it's just new to me, I guess. We're going to be using this new technology called Poll Everywhere. And there's 3 different options for how you can participate. Heather just put in a link over in the chat. You can click that link and you can answer this question right in there. And my question for you is, how do you currently teach PCR in your classroom? Number 2, you could text Diane Sigalas 608 to 37607. And then you can answer that same question there. Third option for you, because we like to differentiate and make sure everyone has access to it, you can take your phones and you can scan this QR code, open up your camera, point it at your screen. You'll be able to participate in with us right now. And if we go back, you'll be able to see that people are answering and that their things are going to pop up right here so that we can, like Tim said, still have this conversation going, even though it's remotely. All right, so we use some simulations, definitely. Some videos, absolutely. Introductions to gel electro, definitely, same with me. Slides in a PowerPoint, that is 100% the way I used to go. Learn genetics, I love that site. A quick lecture, I hear you. I hear you. Some simulations, time's running out. All right, so slides in a PowerPoint. Learn Genetics, fantastic resource, love them. Watch a simulation, some paper models. I get you. This is exactly what I did too. You know, a little bit of Mr. Anderson, anyone? Some YouTube videos. This is what I used to do. But what I want to show you is, well, here's my old slide. Everyone with me? This is our slide in our deck? Yeah. Here's my question though. With this slide, with those simulations, with that quick YouTube video, with pointing up to your board to where that poster is sitting, how well do the kids know the topic? I'm talking really know, not the owls regurgitating, really know the topic. So come with me. Let me show you a different way. Let me show you a way that I will never turn away from again. Let me show you a light, a different option, a different way that absolutely revolutionized how I teach PCR. So I just want to go over a few things to make sure that we're all on the same page. We all use this probably. It is our picture of our nucleotide. We talk about the different numbers of the carbons and where everything is. We make them memorize this. Now let's make them use it. So right here is your nucleotide. So with your nucleotide, I just want to orient everyone in case this is the first time that you've ever seen these nucleotides before, just so that you can see how these nucleotides are set up. Again, it depends upon where you are in your curriculum. With PCR, I've played with these nucleotides a few times so that even my 17 and 18-year-olds come back and answer the questions. But again, let's just make sure that we all see where we are. So this side here is going to be our 5'. I always tell them, notice how it has that hole that's missing. I tell them to think about the 2 phosphates that fell off so that our nucleotide is ready to bind. And here you have your 3' side where your hydroxyl is. Notice also that it looks literally like arrows. So it highlights again that 5' to 3' nature of the structure and how things are going to work. That's the thing with these kits. It's all about colors and shapes and the repetition of these colors, how they go through all of the different kits that you're going to get, how these themes come back again and again and again. It helps drill it into our kids. Modeling equals mastery. The more times they model, the better they're going to master the content. So we can get ready to play. I hope everyone took some time to pre-assemble. I saw Heather put into the chat just before the double-stranded structure that we're going to be working with today. Here we have it right here. So I hope you took some time to put this together. If not, here's our set of nucleotides up here on the screen. Also, again, Heather put it in the chat so that you can assemble your double-stranded piece of DNA. With this, some options for you. When you're doing this in the classroom, you can always save time by pre-assembling these. So if I'm running a whole PCR reaction, I'll definitely pre-assemble these. The other thing that I'll do down the back, you see how where the DNA nucleotide, where that round shape is on the bottom of it, I'll put a piece of tape right down so that even my clumsy little half-awake seniors in the morning won't break my double-stranded DNA structure, so that they won't ruin our whole experiment for the day or our whole modeling activity. Right, so today we're going to model. Model 2 different cycles of PCR. So what we're going to do to make this work with the kit that we have, I know it's not perfect. It's okay, Tim. We're going to use RNA nucleotides. So we're going to use these nucleotides. Why my little slide got a little messed up, I don't know. But what you need to do is you need to make 3 sets of each of these nucleotides. So the structures for the nucleotides or the sequence for the RNA primers that you're going to make are right there. And you want to have your nucleotides separated off in little, your DNA nucleotides, your free nucleotides, you want to have them off to the side in some separate piles. So while you guys are making your RNA primers, what I want to do is I want to show you what it's going to look like in the big biotech kit. So the first time that you're going to run the PCR reaction or modeling activity, you're going to be using, it's a strip of primers, DNA primers. You can tell because it has that same circular bottom on it. And it comes with a sticker sheet. So you can put the stickers on so that the kids can still match colors like they're used to. What this does is it's larger. It's better for tactile learners, so they can't, less to lose, it's less to break. It really introduces them to the topic in a safe way. As you move forward and you do some of our supplemental activities, you're going to use, trying to not have a shine, you're going to use just plain gray nucleotides. And what this enables you to do is it enables you to design your own primer. And that's a great conversation that you can have with your students. How can you figure out what primer works for what you're looking at? It's a great conversation. We'll talk about it a little bit in a few slides. It's also very timely when you're talking about PCR tests for SARS-CoV-2, really defining primers to look for a specific sequence or a specific RNA segment in terms of the SARS-CoV-2 virus. So some different strategies for you as you guys are finishing up your 3 and 3, for a total of 6. Differentiation possibilities. My kids that are a little higher, I don't always give them a fully formed set of double-stranded DNA. I don't always give them the primers already made. 1, depends on how much time I have. 2, it really depends on my kids. Some of my kids, you know what, they need more things to work on with their hands. So having them spend a little bit of time lets me address something else in the classroom as I'm going through it. So you could pre-assemble, you could not pre-assemble. You could figure out if your kids are capable of doing that or not. So there's a bunch of different options for you in terms of timing-wise with that. Because what we're gonna do now is we are going to use that double-stranded DNA, and we are going to model how we are going to make our DNA, or how we're gonna do our PCR reaction. So what I'm going to do is, to help you guys out, I'm going to switch my display that I'm sharing with you guys. So here we have our double-stranded set of DNA. And what we're going to do is we are going to denature our DNA. Then you'll be able to take your RNA primers, remember they're symbolizing our DNA, and we are going to attach them or anneal them onto our strand. So that's our second step of PCR. We denature, we anneal. From here, we're going to extend. So we're going to continue growing onto the 3' end in the 5' to 3' direction. So we're going to continue adding nucleotides to our growing strand. Okay, nice and simple. So we denature, we anneal, and now we're going to use our free nucleotides to extend out. All right, so I helped you through cycle 1. What I would like you to do is, can you, now that we've done cycle 1 together, can you now denature, anneal, and extend to do cycle 2? Well, while you're working, what I can do is I can get back over to here and I can show you the beautiful stop motion animation. This is of the full size kit, so you can see it's definitely larger. We made a smaller sequence because, well, we only have about 30 minutes. So we're definitely going to do a smaller sequence, but this is what the large sequence will look like and how the kids will work through PCR with the Biotech Kit. So you can see how much space PCR ends up taking on your lab benches. Anyone that's brave enough, raise your hand. Let me see what you did.
To show you my work.
Yeah, let's see.
I have my sequence pre-assembled.
Good.
I did not. I was
Oh,
starting to put it together very quickly using Heather's sequence. I didn't see the 3' in front of the structure that I was making. So when I was all done, I realized I had the polarity wrong. So therefore, I didn't get started here, but I'm on here just to say that this is terrific. I wonder if you want to comment on why you're using RNA primers.
We are
in your
using
student
RNA
modeling pack.
primers for this, Tim, because we're working within the confines of our nucleotide student modeling pack. So
Okay.
not all models are perfect, but that's all right. We are going to use what we have and we're going to make it work. We're going to improvise and it's going to be fantastic. So we're using what we have in our nucleotide student modeling pack to model an imperfect situation.
I agree completely with that approach. It's just that I wasn't sure it was clear to everyone else that you were just using that minimalistic student modeling pack. Okay.
We have another person who's raised their hand. Corrine, I'm going to go ahead and invite you to the stage. Might take a second. I have to remove Tim from the stage as well.
Bye.
Diane, just a time check. I think we have 10 minutes left in our session.
Perfect. Thank you. Hi, Corinne.
Hi. Is this what we were supposed to be doing?
Perfect. There you
Yeah.
go.
If you can lift it higher, we might be able to see a little bit more. Yep.
Is that better?
Yeah. So, yes, thanks for sharing.
Awesome job. So again, like we were saying with Tim, it's a short sequence. It gets the job done. It's going to introduce the topic. It's going to get kids playing with nucleotides, even if you just do this much on a small scale with the nucleotide student modeling packs. And just to make sure that we look again, here's the full-size primers that you'll see within the Biotech Kit. But Corinne, that was perfect.
Okay, thanks.
Woo! What I would like to do is I would like to ask you if you take your phones back out for one last poll everywhere question. I want to see what questions do you think your kids would ask after going through an activity? Awesome, definitely. What happens to all the primers? That's a great question. And a lot of times, some of these questions that my kids were able to start asking about, I never had with the former way that we did it, just having it up on the as a slide in my PowerPoint. Why doesn't it make the same size piece every round? Absolutely. How would we separate the DNA? Perfect. That leads us right into usually our next conversation about talking about gel electrophoresis. Awesome. Again, why would we do this? Perfect. That's a nice one. How do we get the primers made in the first place? So with that question, I'm going to skip to our next piece that I want to show you about, because the main piece of PCR is great. I love that activity so much. But this right here, this is fantastic. And it opened up so many questions for them. It made them kind of extend where they are with how PCR works to the next level. Can you use PCR to find something in the genome, find just one change, which is what ARMS PCR does. ARMS PCR is a way to find a single mutation or a single point mutation within the entire genome. And one of the pieces that it's used to detect is sickle cell anemia. And when you think about it, how many times do we go back to the story throughout the year as we're teaching all different types of topics? Here we can go to it again. So it's a topic that the students are familiar with. It's something that they can make connections with from prior knowledge that you've already gone over. But now you can talk about how you're going to design primers specifically to find one letter change. Tim, we can talk about what ARMS stands for in the booth, because we're just going to keep motoring along.
Diane, I was just going to give you a 5-minute warning and just let all the attendees know that NSTA will be sending out a survey at the end of this session. So look for that as well.
Absolutely. So just to start recapping, here are the different things that you can model. Again, these 5. What you didn't see is right down here at the bottom. And these are worksheets that you're going to get with this kit. Again, all the sequences, questions built in to address mastery, questions to make sure that they understand what they're doing and how it connects to other things, questions that connect them back to scientific literacy and scientific history. Just what did my kiddos think? They loved this. And the biggest piece that they said, my favorite comment was, why didn't we do this last year? Because I did this twice this year, one with my older kids, my second year students, and one with my first year students. And when my second year kids said, why didn't we do this last year? At first I got like, well, I didn't make it yet, rats. But when I started thinking about it today, when we approach this year, this craziness that's going to be this year with having to do just virtual simulations or just data analysis, modeling is going to be absolutely essential to making sure our kids understand what's going on. Without it, they're going to be lost in a sea of Excel. And we cannot let that happen. And if you don't believe me, take it from one of my kiddos. I didn't bribe her, I swear.
I just took biotech with Ms. Sigalas this past school year. She asked me to talk about the foam PCR activity that we did in class, and I thought it was so helpful. I'm a very visual and tactile learner, so able to see what was happening within the DNA and in these tiny little microcentrifuge tubes was really, really helpful for me understanding the concepts of PCR. The foam pieces were really tactile and helped me really comprehend how DNA is able to stop itself at a point where you find exactly what you need. It was actually really helpful to me because one day I came across a video of testing for COVID-19, and they were talking about how these are PCR run tests, and they were talking about primers and solutions that you need, and how PCR works and the temperatures that PCR runs at and such. And I was able to comprehend the entire thing and actually explain it to my family who had questions about PCR and how testing for COVID worked. So I found this kit very useful for comprehending those ideas.
So just one last recap. We were talking today about the Biotechnology Kit, item number BTK01, but you were using the Nucleotide Student Modeling Pack. Like we said, it's smaller, it's limited in what you can do with it, but it's still perfect. And when you think about how we're going to be approaching things, I want to think about this student modeling packs as pandemically perfect because they're great for at home, great for individual use, and they're going to still allow our kids access to the material. So it's a big one for you and an at-home kit for your kiddos. So I want to thank everyone for attending. If you have any questions, please feel free to join us in the booth. Like I said before, I feel alone on the biotechnology island. So I would love to connect with people and really make a nice little cohort of a nice little family of people that teach the same kind of topics. So here's all different ways to contact me or to look at what I do at Livingston, and we hope to see you at the booth.